everyone, and welcome to today's webinar, Optimizing Experimental Design for Gene Editing Success, Tips and Tricks to Increase Efficiency and Improve Outcomes. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by MilliporeSigma. To learn more, visit sigmaaldrich.com/US/EN. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Fuqiang Chen, Principal Scientist at MilliporeSigma. For a complete biography on our speaker, please click on the Presenter tab. Dr. Chen, thank you for joining us. You may now begin your presentation.
Hello, and welcome to my webinar on optimizing experimental design for gene editing success, tips and tricks to increase efficiency and improve outcomes. My name is Fuqiang Chen, and I'm a principal scientist for MilliporeSigma. MilliporeSigma is the life science business of Merck KGaA, Darmstadt, Germany, and operates as MilliporeSigma in the US and Canada. In today's webinar, I will share with you my experience in optimizing gene editing experimental design. First, I will discuss target site and nuclease selection, and guide RNA and RNP complex preparation in the context of CRISPR gene editing systems. And will then focus on HDR gene editing. I will explain why the distance from cut site to edit site is critical to HDR editing efficiency, and give you some tips on how to optimize donor design, how to enhance HDR editing with small molecule NHEJ inhibitors or other strategies, and how to handle cell culture and transfection. And at the end, I will also provide some tips on editing analysis and single-cell cloning. Gene editing is a complex process. It involves many aspects of cell biology, and therefore success depends on many variables. And some of the variables are difficult for us to manipulate with our current knowledge, but some we can optimize in our experimental design to increase the chance of success. First, as a gene editing researcher, it is important to bear in mind that gene editing relies on cellular DNA repair pathways, and the complexity of these pathways in eukaryotes often poses constraints for us to achieve desired editing outcomes. For instance, NHEJ and homologous recombination are two major competing pathways for DNA double-strand break repair in mammalians. And as we know, NHEJ is dominant over HR most of the time, and this is a challenge to HDR editing. Fortunately, we now have a certain ability to manipulate these two pathways to improve editing outcomes. And this ability will continue to advance in the future. Before embarking on a gene editing experimental design, it is also highly recommended to know the structure of your gene of interest in as much detail as feasible. This will help you to find the best place to introduce indels for gene knockout, to tag the gene, alter the gene function, or regulate the expression. The conventional approach for gene knockout is to introduce indels in one of the early exons that encodes the protein and terminal region to create friendship mutations and premature stop codons. And this will lead to nonsense mRNA decay. And for more complex gene modifications, a good knowledge of the gene structure will certainly be very useful. Why CRISPR? After over 3 decades of collective efforts, we now have several gene editing platforms to choose from. There are meganuclease, gene finger nuclease, TALEN, and of course CRISPR. Which platform is the best for you will depend on your specific circumstances. But generally speaking, CRISPR is currently the dominant editing technology, and simply due to its intuitiveness in target design, its flexibility, and its rapid improvement in many aspects. In contrast, meganuclease, GFN, and TALEN all require complex protein engineering for each target site, and their improvement is much slower compared to CRISPR. The physical form of a nuclease you introduce into a cell can be critical to your editing outcomes. In the past, we used plasmid DNA, mRNA, or viruses for delivering a nuclease. And now with CRISPR, we have a new form of delivery in RNP complexes. For most gene editing applications, we recommend using RNP complexes or mRNA because they have no risk of random integration in the genome. They can help to reduce off-target effects, especially RNP complexes, and they are less likely to cause damage to cells in electroporation. In contrast, plasmid DNA and viruses pose random integration risks and have higher off-target effects due to their overexpression or long-lasting nature. Furthermore, some cell types, such as T cells, GeoCat, and A549, are highly sensitive to plasmid DNA in electroporation, especially when large plasmid vectors are used. Target selection is an important step in CRISPR gene editing experiments because of the concern over off-target effects. In a complex genome, such as the human, chances are you will often find similar sequences for a given Cas9 binding site. Fortunately, there are several computational programs to help you identify potential off-target sites. For example, the COSME program is quite popular for searching potential off-target sites. And you can also use an in vitro cleavage assay to identify off-target sites. However, the actual off-target effects in an editing experiment depend on a wide range of variables, such as the number of mismatch position, nuclease mismatch tolerance, nucleosome positioning, local chromatin structure, and epigenetic status. Some of them are difficult to predict, if not impossible. If you are really concerned with potential off-target effects, we recommend using a nuclease with balanced activity and specificity. Some engineered Cas9 variants are well balanced between activity and specificity, but they tend to be more variable in activity across different target sites, and therefore you may need to test more than one high-fidelity Cas9 variant for a given site in order to find an optimal one. On the other hand, RNP complex delivery can also effectively reduce off-target effects. And you can also use truncated guide RNA spacer length or chemical modifications on the spacer sequence to reduce off-target effects. But in general, if you use RNP complexes with a high-fidelity nuclease, off-target sites with 3 or more mismatches are generally not a concern. Target accessibility is a major determinant of editing efficiency. However, target accessibility is a complex issue and not well understood. There are too many factors that can affect target accessibility, such as the GC content and distribution, nucleosome positioning, and epigenetic status. The potential secondary structures when the target DNA sequence is converted to the guide RNA sequence. You may use a computational program to predict which guide RNA may have the best activity on a given genomic region. But we recommend performing experimental tests if you have a limited number of candidate guide RNAs. And as illustrated here, some closely positioned guide RNAs can have very different activities, even though they may differ by just 1 or 2 base pairs. Why use a synthetic guide RNA? After you have selected a guide RNA design, the preparation of the guide RNA reagent can have a major impact on your editing efficiency. Traditionally, we used in vitro transcription to produce guide RNA for transfection. And now chemically synthesized guide RNA is widely available and affordable. And we strongly recommend using synthetic guide RNA and not in vitro transcribed guide RNA in cell editing experiments. Synthetic guide RNA has 2' O methyl group and phosphorylsilyl linkage modifications on both 5' and 3' ends to increase resistance to nuclease. In addition, chemical modification on the guide sequence has potential to improve editing specificity. In contrast, in vitro transcribed guide RNA is less stable. It can be immunogenic to certain cell types if the 5' triphosphate group is not removed. Furthermore, T7 RNA polymerase requires a 5' G for transcription initiation, and this can limit guide RNA design flexibility. How do you prepare RNP complexes for transfection? Can also have a major impact on editing efficiency. And here are some observations we found to be useful in preparing Cas9 RNP complexes. You want to maintain about 300 millimoles of salt in the assembly to keep Cas9 protein stable. And you want to use excess guide RNA to protein in mono ratio. And for most applications, we recommend a mono ratio of 3:1. But for difficult to edit sites, the ratio can be increased to 10:1. And keep RNP complexes on ice before transfection. And if you use plasmid DNA for transfection as donors, prepare the plasmid DNA in a separate tube. To protect guide RNA from degradation by residual nuclease, as you may have personal experience, a large amount of nuclease is commonly used in plasmid purification. And when doing the transfection, mix the cells with RNP complex first, and then with plasmid DNA, and transfect immediately. But if you are using synthetic oligos as donors, you can combine the oligos with the RNP complexes in a single tube for transfection. I will now focus on optimizing HDR gene editing experimental design. HDR editing can accomplish all precision gene editing needs, from changing a single base pair to integrating a whole gene, and promising improvements to increase efficiency and product purity will make this tool even more useful in the future. And besides precision editing, you can also use HDR editing to enhance your total gene knockout efficiency. And as illustrated here, you can simply add an oligo donor that encodes a small friendship deletion or insertion into your transfection, and you will capture indels not only from NHEJ events, but also from HDR events. And the result is a much higher total gene knockout efficiency compared to the conventional approach. And this approach is particularly useful when you encounter no knockout efficiency at some locations. What are the important factors that need to be considered when optimizing HDR editing experimental design? The first one is the distance from cut site to edit site. You want to find a cut site as close to the edit site as feasible. However, this can be challenging, as you may not have a close-by PAM for nuclease target binding. The gene editing community has been trying to address this issue by developing Cas9 variants with relaxed PAM requirements, or Cas9 orthodox that use different PAMs. And besides the cut site to edit site distance, donor design, HDR enhancement strategy, cell culture condition, and appropriate controls also need to be carefully planned. And for controls, we recommend at a minimum using a donor-only transfection control to ensure results can be interpreted. As I have just pointed out, the distance from cut site to edit site is critical to editing efficiency. In general, we recommend keeping the distance to within 20 base pairs. Based on our experience, when the distance exceeds 20 base pairs, the efficiency may decline drastically as the cut site is farther away from the edit site. While HDR editing efficiency declines as the distance from cut site to edit site increases. And here is a model to illustrate the relationship. After DNA double-strand break and 5' end resection, the chromosomal DNA 3' end can use the donor as a template for extension and copy the modification from the template to the chromosomal DNA. And after this extension step, there is an end capture and flap processing step. And during this step, the modified DNA segment could be retained, and this will lead to non-productive HDR editing event, but the modified DNA segment also could be clipped off, and this will lead to a non-productive HDR editing event. And the probability of a non-productive HDR editing event increases as the distance from cut site to edit site increases. And besides this DNA repair step, other factors may also contribute to lower editing efficiency as the distance from cut site to edit site increases. Single-strand DNA oligos are the most effective donors for small precision modifications by HDR, and they have been widely used for over a decade. They can provide a high copy number, have no risk of random integration, no cellular toxicity, and are easy to manufacture. When designing a single-strand oligo donor, we recommend using a 50 to 60 nucleotide homology arm on each side, and using silent mutations to prevent recurrent cleavage after a HDR editing event if the editing itself does not alter the original nuclease binding site to be unusable. And most importantly, the oligo strand orientation must follow the positional relationship between the edit site and the cut site. And I will illustrate this relationship in the next two slides. The first positional relationship is the edit site is upstream from the cut site. In this configuration, you want to design a same-strand oligo as donors. And as illustrated here, after DNA double-strand break and 5' end resection, the chromosomal DNA 3' end can use the same-strand oligo donor as template for extension and copy the modification on the template to the chromosomal DNA. And if you use an antisense strand oligo donor, the chromosomal 3' end cannot copy the modification on the template to the chromosomal DNA unless the 3' end is resected to pass over the intended modification site. In most systems, 5' end resection is much more robust than 3' end resection after a DNA double-strand break. The second positional relationship is the edit site is downstream from the cut site. In this configuration, you want to design an antisense strand oligo as donors. And if you use a sense strand oligo, the editing efficiency will reduce drastically. And the reason is the same as in the first configuration. You can enhance HDR editing efficiency by using small molecular inhibitors to NHEJ repair proteins, especially the inhibitors to the DNA-dependent protein kinase catalytic subunit. Nu-7441 is a potent DNA PK inhibitor, but new improved inhibitors have been shown to be more effective than Nu-7441. The inhibitors to ligase 4, Q7080 heterodimer, and DNA polymerase Q also can be effective in certain settings, but they are in general not as effective as DNA PK inhibitors. However, when using these inhibitors, one must exercise precautions, and we recommend using the lowest effective concentrations, reducing the exposure time by washing off the inhibitors as soon as feasible, and using IMP complexes and oligo donors to shorten the editing window and reduce the inhibitor exposure time. Besides small molecular NHEJ inhibitors, you can also use other strategies to improve HDR editing efficiency. For example, you can use ubiquitin variants to inhibit p53 binding protein 1, which is an antagonist to HDR. You can also fuse Cas9 with dominant negative NHEJ protein mutants, with cell cycle regulated proteins, with N resection stimulating proteins, or with proteins that can carry modified oligo along to the Cas site. However, the effect of these strategies may vary across different cell types. Cell transfection is a key step in a gene editing experiment and needs to be carefully planned. First, you need to consider whether it is necessary to grow a single-cell clone as starting materials for editing, because some immortalized cell lines are highly heterogeneous. And for transfection reagents, again, we recommend using IMP complexes or mRNA for nuclease delivery, and using oligos or long single-strand DNA as donors, especially if your cells are sensitive to transfection or are limited in amount. Grow the culture to log phase before harvesting for transfection, and select a transfection method that can balance transfection efficiency with cell viability, and again, use appropriate transfection controls. Here are some tips we found to be useful for handling cell culture before and during transfection. First, you want to use a culture that has been recently thawed and is at a fast-growing state as starting materials. Avoid using the culture after 1 month of pasture. and harvest the cells for transfection when the culture has an optimal cell density. For suspension culture, the optimal cell density is between 0.5 and 1 million cells per ml. And for adherent culture, it is between 60 and 80% confluent. Wash cells with Hank's balanced salt solution, or PBS, at least twice if you are using mRNA or RNP complexes, because cultures may have high levels of nuclease in the medium and on the cell surface, and try to return transfected cells to normal cultural conditions as soon as feasible. After transfection, you may want to know as early as possible if the editing is successful, and this can be done by conducting an assay 2 to 3 days after transfection. For HDR gene editing experiments using oligos as donors, we recommend purifying the genomic DNA using a bind and elute genomic DNA purification kit before target amplification and sequencing. Do not use quick extra for amplification, because quick extras contain residual oligos and can lead to unreliable results or false positive. And as illustrated here, residual oligo donors can act as primers in PCR amplification and produce PCR products that carry the modification and interfere with your assay. However, if you use a plasmid DNA as donors, you will not be able to remove the residual plasmid DNA donors using a bind and elute purification kit, and you will need to design a junction PCR assay to detect target integration and avoid amplification of residual plasmid DNA. For single-cell cloning, we recommend using flow cytometry for single-cell sorting for most applications. Flow cytometry sorting is very efficient in colony formation and generally does not produce doublets. But some cell types are sensitive to flow cytometry mechanical stress and have very low percentages of colony formation after sorting. And for this kind of cells, we recommend using limiting dilution to conduct single-cell cloning. And generally, a 0.5 cell per well dilution is a good compromise between colony formation percentage and doublet percentage. In some applications, HDR editing events can be rare, maybe because the edit site is too far away from the cut site, or the cells have low HR activities. To isolate such rare HDR events, we recommend conducting pool screening first to identify a positive pool before attempting single-cell cloning, and each pool can range from 50 to 100 cells. However, if your HDR edits contain a fluorescent protein tag, use the power of flow cytometry sorting to enrich positive cells multiple times and expand the cells for a few days between each sorting. In summary, I was trying to convey to you that optimal experimental designs can help to alleviate the constraints of DNA repair pathways on gene editing. And I have emphasized HDR-mediated precision gene editing as a versatile tool and shared with you some observations and tips for increasing HDR editing efficiency. And these include placing the cut site close to the edit site, using a correct oligo donor design based on the positional relationship between the cut site and the edit site, growing a rapid dividing cell culture for transfection, and judicial application of small molecular NHEJ inhibitors. As we gain more knowledge on cellular DNA repair systems, we can make HDR gene editing even more versatile and efficient in the future. Thank you for attending the webinar.
All right. Well, thank you so much, Dr. Chen, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some questions coming in here, so we'll get started with this question. Dr. Chen, this question asks, does the guideline for the distance from cut site to edit site also apply to plasmid DNA donors?
Thank you for the question. Yes, it also applies to plasmid DNA donors. And the distance effect with the plasmid donors can be even obvious, because in plasmid donor, the copy number per cell is usually much smaller than the oligo donors. So you want to try to find a cut site very close to the beginning of the edit site.
Great, thank you. All right, next question asks, how do I calculate the molar ratio of guide RNA to recombinant Cas9 protein in RNP complex preparation if the Cas9 protein is supplied in microgram concentration?
Yes, most consumer suppliers of the Cas9 proteins are in micrograms concentration. And so, you need to convert the micrograms to picmoles in order to make the good calculation of the ratio between recombinant protein, Cas9, and the guide RNA. And for example, if you are using SP-Cas9, and the protein molecular weight is about 160 kilodaltons. So if you divide 1 microgram by the molecular weight, you end up with about 6.2 picomoles per microgram of the protein. And if you use 5 micrograms per transfection, then you will need about 100 picomoles of a synthetic RNA to achieve a 3-to-1 molar ratio.
So, and if you constitute the synthetic RNA in 100 micromolar concentration, that is usually the manufacturer will recommend. In that situation, 1 microliter of that RNA is equal to 100 picomoles of picomoles.
Great, thank you so much. We have time for a few more questions here, so we'll continue. This next question asks, can I use synthetic guide RNA with Cas9 mRNA for transfection simultaneously?
Yes, you can use, you can combine the synthetic RNA and the Cas9 mRNA in a single transfection. It works very well, although it's not like you are not pre-complex them in the RNP situation, but it works very well. And usually you want to, you can calculate, in this case, you can calculate the molar ratio between guide RNA and Cas9 protein. But a general rule of thumb is that for each microgram of Cas9 mRNA, you may use about 40 to 50 picomoles of synthetic guide RNA. And in general, you are using a little bit more of the guide RNA. It does not hurt the experiment. Usually will have the positive impact on editing efficiency.
Great. All right, it looks like we have time for one more question here, so we'll go ahead and wrap up after this one. This final question asks, what are the pros and cons of HDR gene editing compared to base editing?
Yeah, so this is a little bit of the question is still being debated in the community. But from my personal opinion, the HDR editing is a very versatile one. It can make both transition and transversion single base modification, and also can make a large integration. But HDR editing does need double-strand DNA breaks, and also needs exogenous DNA donors for the repair or for the integration. In terms of the base editing, it's generally restricted to a single base pair transition modification. And also there's some limitations because you will have unwanted editing on adjacent cytosine base. But base editing does not require, in principle, does not require DNA double-strand break and does not require exogenous DNA donors. But in practice, you will get the double-strand breaks and also the indels byproducts.
All right, great. Well, thank you so much, Dr. Chen. That will conclude our session for today. Do you have any final comments for our audience?
I would like to thank the audience for attending the webinar again.
Great. Well, thank you again, Dr. Chen, for your time today and your important research. We would also like to thank LabRoots and our sponsor, MilliporeSigma, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.